Really appreciate everyone's input from that first session. One of the things that we have always been keen to do as part of this network and as part of this community of people is make sure we engage with everyone, and we think that the industry view is really important. And that's through all of the equipment and the assays that are being developed, all the way through the ability to commercialize and do something with your research. And we're going to hear about a lot of those things in this session. I'm super grateful to everyone that's participating in this. And without further ado, I'd like to introduce someone who's probably familiar to a lot of the guys here on this call, Dimitri, who has been in scientific equipment and services and business development for his professional career through Izon and through NanoFCM. And he joins us today as founder and CEO of Vesiculab. And he's going to talk to us about the research workflow and how we can look to improve that from production all the way through to our assays. So Dimitri, welcome. Take it away.
Thank you very much for the introduction, Matt. I'm just going to share my presentation. Can you all see that? Yeah. So yeah, thank you again for the introduction, Matt. So yeah, today I'd like to present the activities of my new company, Vesiculab, which aims to improve every aspect of the EV workflow, really from production to functional assay. So very quickly, I present what is Vesiculab. It's a very young company. The activity started only in June, and based with Village, so north of Nottingham, very Midlands. And we offer reagents and consumables for EV research. So really all the products that are bolting on to the equipment that you guys already have in the lab, the analyzers, the bioreactors, and the microscopes. It is active worldwide, but it doesn't mean that I don't want to be more involved into the local research community. So it's great to be here. So very worldwide, as you can see, we've been already busy, and we have users across 3 continents for now, 5 soon. And I'm really looking forward to add you guys in the Midlands to that very long list. So what exactly we're doing is that we're bringing solutions into the portfolio that are helping each aspects of the EV workflow. So really running a small survey in August, it was clear that there are still obstacles to a smooth pathway from production of EVs to look at their functions and how they can be used for diagnostics or therapies. So that's why the portfolio is going to assist on these aspects. And the philosophy is really trying to make the best use of each of these instruments that you guys have been presenting, from NTA, flow cytometry, microscopy, etc. What's important is really to reduce the time to results. It's been evident over the many years of activities that it was taking a long time to get to even isolate EVs that were workable for functional assays, and that can really slow down not only a whole piece of research, but even doing in a PhD. So the portfolio is starting with just a few products, but it will be growing to cover all these aspects and improving each of them. And on the personal level as well, I want to continue what I've been doing for many years. Obviously, Andrew David can second that. Fostering academic collaborations, so taking part to grant applications, for example, to help recruit PhD students. Can be more limited projects. I've been done all that in the past, and I'd be very pleased to do that again through Vesiculab, and overall really contribute to the Midlands scientific industry as the company develops, there will be obviously more science research and as well as manufacturing going on in Nottingham. So I'm going to quickly show the products that are already in a portfolio. The first one to be released was Vesiculab CD63 GFP, a reference material. It's an EV isolate from HEC cells, and that is isolated by TFF and purified by size exclusion chromatography. It can be used really as on many platforms as part of an orthogonal validation process, and that's quite important because each of the techniques that are used for analysis needs to cross-reference to a material that is known as being EVs and has all the biomarkers that makes a real EV basically. So and the CD63 GFP positive aspect is very important as well for any assay which is based on fluorescence. It can be NTA, it can be flow cytometry, it can be as well confocal, making sure that there's the ability to detect a signal before just going to the sample and see what's in there. The second product is Vesi dye LMB 600. So it's a lipid dye that is intercalating into the lipid bilayer. It's not specific to EV. It's really the lipid bilayer of any membrane, really around nanoparticles or larger objects. But it's really perfectly suited for EVs. It stains all EVs, so it's not really specific to a specific marker, but because it's water soluble, it doesn't form micelles or aggregates that tend to pollute the measurements on any techniques and functional assays actually. So it is going to be compatible with the techniques I mentioned before. So fluorescence NTA, like the previous speaker. It's quite important actually to assess that the particles that are measured are EVs, not just impurities that could be in the medium. It works as well with fluorescence MRPS and flow and nanoflow cytometry. And for the functional assay, then it is a very, very good dye for functional assay because it has a very long lifetime for the intensity of fluorescence. So you're able to do measurements over long periods with the fluorescent intensity lasting up to 7 days. And the third product that was introduced very recently is called Vesi Micro. They are a small spin column made with a size exclusion chromatography gel, and they are used for a very fast and efficient cleanup of samples before they go into these analytical techniques or functional assays. So it works on the principle of size exclusion chromatography. So it means that the largest objects are going to be elute. Anything over the cutoff of 35 nanometers is going to be eluted, and the smallest impurities are going to be stuck on the gel. And the smaller impurities are chemicals, like for example the dyes that are used to stain EVs, or they can be antibodies as well. So it's really for for sample size of 50 to 100 microliters volumes, and it's because it's a 2 milliliter collection tube format, it's possible to use it in centrifuge. So it's really basic equipment. Best to use it on the swinging bucket centrifuge. These are widely available, and in this way, it's possible to scale up to or even over 100 samples at a time. So it's possible to envisage very large studies, which is quite important because at the moment it's very difficult to base a strategy on gravity, size exclusion chromatography columns alone. So really, in summary, I'm very, very happy to be here because it's really a way to reconnect and again reiterate that I'm very, very, very keen to collaborate with you guys locally and really help the field of EVs to blossom in the Midlands. And if you think about applying for grants where you need a knowledgeable SME to contribute, then don't hesitate to contact me. It's the same if you have if you think that you have an IP or something that you would like to commercialize in this field, then it's something I can have a look at. And you can count on me for the next event that hopefully will be in person. Really looking forward to it. Thank you very much.
Brilliant, thanks, Dimitri. Yeah, I think we've all described some of the challenges around EVs, and I think some of these products are going to be really useful going forward. I had a quick look. Can you see any questions, Lucy?
No, I was just going to say, I think because we've run over slightly there, I think in the interest of time out, we'll move on to the next presenter. And Dimitri, if anyone's got any questions, I'll put them in the chat for you, if that's okay.
Thank you.
Thank you. Thank you very much.
Brilliant, thanks. So next up, it's my pleasure to introduce Rob Tempest from NanoFCM to give us insights into the work they're doing with their nanoflow cytometry. Rob, looking forward to hearing from you. Take it away.
Thank you. Just make sure you can see this. Is that all okay? Great. So yeah, my name's Rob from NanoFCM. And yeah, it's a pleasure to be part of this first meeting and the launch of the EV network for the Midlands. What I'm here predominantly to talk to you about today is the NanoAnalyzer, which is our platform for comprehensive EV analysis and characterization using a technique called nanoflow cytometry. So who are we as a company? NanoFCM, really what we are as specialists in nanoflow cytometry and in EVs. So we actually have multiple people within the company who have their PhDs in EV work and really years of experience in nanoparticles in general, but EVs traditionally has been a huge part of our business. And predominantly what we have is the NanoAnalyzer, which is our flagship platform. We're actually also based in Nottingham at the Medicity on the Boots site. You can see here in the bottom left. So we've already been active in the local community, taking part in the things like the flow events and things, but really keen to build on that relationship with people here. The company itself is actually a spin out from Xiamen University in China. And that was back in sort of 2013, but since around 2018, the UK has been the headquarters for all operations in Europe, as well as in North America as well. So we're very central to everything that's going on within the company. So in terms of our actual technology, what is it and how does it work? So we do nanoflow cytometry, where a lot of people have been using conventional flow cytometers to try and look down at this nanoscale. Realistically, those cytometers are built to look at cells, right? Cell-sized events. So there's been a lot of work put into sort of dragging them down to this lower end. But realistically, we don't see much convincing work below 150 nanometers. And even then, we're seeing working very close to the noise or relying on standards made of things like polystyrene that aren't really reflective of biological materials. What we have is a system that purely focuses on the nano scale. So we work for biological particles in the range of about 40 to 1,000 nanometers. So that allows us to really get a clear picture of what's going on at the EV level, as well as working in these other areas here like viruses and lipoproteins. So what's actually going on in the NanoAnalyzer? If you sort of follow along with the video in the bottom right, we're loading as little as 10 microliters of sample at a time, and actually less than 1 microliter is consumed in a standard sample acquisition. So very good for those sort of precious samples. That's then boosted through the system with a highly controlled sample pump, where we actually refine that sample stream down to a core stream of 1.4 microns. So that allows us to get really reliable single file movement of particles through the laser. And also compared to standard flow, we work at a lot slower flow rates. So that means that each particle has an extended dwell time in front of that laser, and we can gather more light information, which is what allows us to have this increased sensitivity, which we kind of then build on with these really sensitive single photon counting detectors. You can see a kind of rough idea of the output here, but really of interest is on the left here, which is what we call our event burst trace. So each one of these lines is a particle passing through the laser. And just to go into that in a bit more detail, so we have those event burst traces here. So these are simultaneous measurements where in blue here we've got side scatter. So that's going to be related to the size of our particle and as well how we calculate the concentration. And then here we've got a FITC fluorophore as well. We do have 2 fluorescence detectors, but just for this example using 1. So we've got a couple of situations really. We have a side scatter signal with no corresponding fluorescence. So that's going to be a particle that's over 40 nanometers, but doesn't have our labeled antibody to our target of interest. And then we might have simultaneous measurement of those. That's going to be essentially a positive event. And if you look at the dot plot here, simply by gating across the y-axis, we can define those 2 populations and get an exact percentage based on a particle by particle measurement rather than a bulk analysis of the percentage positivity. And something important that we do for maybe, I'd say over 95% of our EV work is we use a side scatter based trigger. So any particle displayed here has to have that side scatter signal, therefore has to be over 40 nanometers. So if you had something like degraded membrane or free fluorophore in solution, it's not going to be big enough to generate that side scatter signal. So it gives us a really nice robust data set that doesn't have those false positives counted. So in EV research, what are we really looking for? We might just want simple information about the size and concentration. We might be interested in disease state differences, so specific biomarkers we expect to change expression profile in different circumstances. We might be using our EVs as a therapeutic vehicle, so we might be interested in our API cargo. How efficiently is it being loaded in? How much is left in solution afterwards? We might want to know about this ratio of non-desired particles. So it's been mentioned previously, especially in Vicky's talk, about co-isolation of other contaminants, such as things like lipoproteins. So how much of our sample is what we're interested in? And the impact of our isolation and production techniques. So we may be running a 3-step isolation process, but we only get a 1% increased purity from our last step. Are things worth our while, and what are we really seeing at the end? So traditionally, people have used techniques like NTA and DLS, things based on Brownian motion and making some assumptions about that movement and relating that to the size and concentration. And the gold standard for sizing would really be electron microscopy. And then that really only gives us information about the physical attributes of the particles. Whereas when we want to look at that phenotypic information, typically people have turned to things like Western blotting or ELISA's bulk measurements, which tell us about our sample as a whole. So really, the workflows are built around multiple different instruments and bringing all that information together at the end. What we think we provided in the nanoanalyzer is a single instrument solution to that. So it's multi-parameter characterization of your EVs. So straight away, without any labeling, you'll get your sizing and concentration down to 40 nanometers. You get a nice size distribution profile like this, as well as your exact concentration. And then we might want to build on that with something like a purity assessment, kind of that's been discussed already, by labeling the membrane and comparing to other potential protein aggregates that might be in there, for example. And then we might want to label a specific protein. Typically, this is going to be tetraspanins when we're looking at our characterization, but any marker of interest that we might want to look at. Similarly as well, that could be instead, it could be a nucleic acid stain, for example. So any cargo that we can get to fluoresce, we can analyze. So what that gives us here is this dot plot where we've got our membrane label on the x-axis and our, let's just say CD9 for ease, on the y-axis. And what we've got is 4 populations where in blue, these are only side scatter positives. So they have a side scatter signal and are over 40 nanometers, but no membrane positivity or antibody. So you could infer that then these are our non-EV contaminants. We've got our membrane positive only, antibody positive only, and then importantly, that double positive population. So the co-localization of those 2 signals, which tells us about, in this case, membrane bound EVs that are also positive for CD9. And we can go into that in a bit more depth with things like size overlays to look at trends between the different sizing and different subpopulations we have. Lots of different analysis options in the software. So in summary, for that sort of technology side of it, what we're doing is concentration and size distribution plus your phenotypic characterization, all in very quick 1-minute run times with a couple of minutes analysis per sample. Then I just thought I'd give a little bit about what we're currently already doing in the Midlands, so the sort of projects and collaborations we're working on. What we can generally offer, obviously our core business is going to be that instrument demonstration and sales. So we are frequently on the road doing demonstrations for people, and then that relates to instrument sales. But also currently a big part of our business is the CRO side of the service. So you just pay on a sample by sample basis for analysis work. And the advantage with that is that you get all the expert advice and experimental planning that comes from all our experience working with these with EVs. And obviously we do all the legwork as well. And we do offer preferential rates for academia. And then aside from that paid service, we do also do collaborative work on a free basis, because either it contributes something to a paper or because it's mutually beneficial, or there's lots of different routes that can lead to that. So just to give you a bit of an overview, we're doing some work at the moment with Keele University, and we have been for a while, looking at things like MSC markers in EV isolates from biofluids. And then we've been doing things like assessment and characterization from patient samples from the local hospital. University of Birmingham, we've had several collaborative projects there. We've actually had a fair few that are non-EV related that I haven't listed here, doing things like magnetosomes, for example. But in terms of AVs, we've done a fair bit there that have resulted in 2 EV publications, as well as one that's in revision at the moment. At Aston, we've had a industry placement, and I think Rachel's talking later today, so she spent a few months in our labs as part of her PhD programme. And then we have ongoing work there, working with our research team. At the moment, we're looking at a comparison project between different analytical techniques, for example. So how does our nanoanalyzer stack up against some of the standard techniques or even non-standard ones? And there was actually a nanoanalyzer installed at Aston a couple of months ago. At Nottingham Uni, I've been talking to Lucy recently, and we're looking to set up a collaboration there whereby students are going to come and join us at our Nottingham site and do some assessment of their EVs, as well as looking at, for example, their current staining methods that they're using. And we have had previous collaboration there with SARS-CoV-2 virus, where we had the instrument in level 3 containment and looking at spike protein. And the final example I'll give you is with Loughborough University. So we have an ongoing collaborative project, a PhD project with them, where they're looking at drug loading of EVs, as well as some liposomal work. And that is in conjunction with us, where they are based at Loughborough, and then they come over to be based in our labs to do additional analysis for their samples. And in Loughborough in general, again, we've had a couple of EV publications through that collaborative work, with several more in the works at the moment. So yeah, that's kind of a brief overview, I would say, of what we can do and what we are doing. If you wanted to reach out to us, we're very interested in further collaboration. From the sort of sales and application side, there's myself that you can contact, and then Ben, who is our head of research, is much more heavily involved in these collaborative works that we're doing, as well as the CRO side of the business if you wanted to do samples on a paid by basis. But yeah, happy to take any questions.
Brilliant, thanks, Rob. And I think it's great that not only you guys are local, but are so supportive of working in with academics and at different levels and different capacities. So I think that's really important for the community and for our ability to fund some of these studies from proof of concept all the way up to kind of clinical work. So
yeah, we're looking to build on that more at the moment as well. I mean, we've been spread all over Europe, and there's still a lot to do focused in this local area where we have such easy access. So yeah, more than happy for people to reach out with us and discuss any projects they might be thinking of.
Brilliant, thanks, Rob. Appreciate that. I think there might be a question, but we can either pick that up in the chat, or we're not too far away from a coffee if people want to connect over a digital brew. So next up is Harry Newberry from Cell Guided Systems, and he's going to have a chat with us about Excellizer and what you guys are up to over there. So Harry, whenever you're ready, take it away.
Yep, thank you very much. I'll just make sure this is working. Okay, is that looking okay?
Yeah, looks good.
Cool. So hi, thank you for having me, and a huge thank you to all the speakers so far. It's been really interesting. So I'm Harry, as you may already be aware, and I'm a research scientist here at Cell Guidance Systems, and I'm also heading the exosome group here, doing various R&D. So I want to talk to you a little bit about what our company offers to the EV field and how we can help researchers. In particular, I want to kind of talk about our ExoLyzer technology and how it can be utilized as a simple validation solution for your research. But briefly, as a company, we offer a range of different products and services, namely our widely cited ExoSpin SEC columns, which are used obviously for isolating EVs from a range of biofluids and various volume ranges. And we try to strive to allow high throughput isolation with these. We also offer a number of different EV-related services, such as NTA, validated antibodies, and most recently, we've produced a line of lyophilized EVs, which are from a range of different human cell lines. But as I mentioned in the title, what I'd really like to talk to you about today and focus on is the ExoLyzer assay. Briefly explain how it works and how it can be used as a validation tool in your EV research. So as a typical workflow for most of you, which will probably understand this, EV studies usually consist of yielding EVs from a range of different sources, such as cell culture media or human plasma, for example, and then choosing a method of isolation, be it ultracentrifugation, or perhaps a combination of different techniques. And then, of course, at this point, it's really important for you to actually validate the presence of your EVs so that prior to further downstream analysis, you can actually validate and know that what you've got and your isolation technique has worked. And of course, lots of common techniques will include things such as Western blotting, which, at least in my experience, and perhaps yours, can be quite time consuming. It isn't really standardized in the field. Quite a lot of questions as to, you know, should I use reducing or non-reducing? How much should I load into the sample, etc. So this is kind of where the ExoLyzer steps in, and I think is a really good tool in your workflow. So the way that it works is very similar to a typical ELISA assay, but ultimately it differs in that it uses the same antibody for both capture and detection. And this is obviously key when working with EVs, as it will only detect vesicles with multiple copies of a specific surface protein, let's say the tetraspanins, so either CD9, 81, and 63. And this is really, really important when working with vesicles that have multiple copies. And so the way that would work is very simple. You would begin by adding a bioattenuated antibody, which would come with the kit, and this will bind with your streptavidin coated plate. After an incubation and a wash step, you would then add your purified EVs. And again, following incubation period, you would then add your europium labeled antibody. At this point, after around about 3 hours workflow, you can read your plate and generate results. And again, it's kind of
Harry, you're sticking. Do you want to go off camera? Maybe we'll be able to hear you more clearly.
Generated using.
Harry, can you hear us?
Notably, as you can see, it demonstrates. Yeah.
Yeah, you're skipping a little bit. Do you want to go off camera and it might come through more clearly?
Is there some connection?
Yeah, we're getting static in each sentence, or at least I am. Maybe it's not everyone.
No, I am as well. Yeah, if you just turn your camera off, probably, Harry, then we'll hopefully get a better connection with you.
And then if not, as you know, I've got your slides, so we can always chop over.
Okay, is it any better now?
We've not lost your camera yet. Do you want to try talking and then see if it's okay? Oh, yeah, we've lost you off camera. Have a go.
Is there anything better now?
Much better. All right, there you go.
It's better, yeah? Excellent. Do you want me to go back a couple of slides or continue?
I think we lost you about half a slide ago, like literally a minute.
Okay.
Yeah, that one. Yeah.
Okay. All right, so just to recap, the way that the ELISA works is it's very similar to a typical ELISA assay, but obviously it differs in that it utilizes the same antibody for both capture and detection. And this is obviously key when working with EVs, as it will only allow you to detect vesicles with multiple copies of the specific surface proteins, such as the tetraspanins, which are commonly used as biomarkers for EVs. And the way that it would work is you would add your bioattenuated antibody, which is supplied with the kits, which would then bind with your streptavidin-coated plates. You would then add your already purified EVs. And then after an incubation and wash step, you would then add your European-labeled antibody. And after around a 3-hour workflow, you would then be able to read your plate and generate results. So quite a nice solution and quicker than a typical Western blot. And then just to show you some previously generated data, we have a graph here which has been generated using the CD9 specific ELISA, which was used upon a number of urine samples from a cohort of 20 patients. And notably, the graph will demonstrate a difference in overall expression of CD9 between the samples. And this is essentially just helping you identify the use of CD9 as a possible biomarker and general areas of interest. And another graph in which the ELISA has been used, this time upon EVs generated from a colorectal cancer cell line. And in this assay, they've looked at the expression of different tetraspanins for this specific cell line. And again, this can help further identify possible biomarkers or areas of research upon the specific cell lines. So thank you for listening. Apologies for any of the connection issues. I'm happy to take any questions, and if you do want to get in contact, please visit the website or email us at the email on the screen, and we'll be happy to support you and collaborate if need be. Thanks.
Brilliant, thanks, Harry. Really appreciate that, and thanks for bearing with us through the inevitable technical glitches that have to happen in any online meeting these days. So appreciate that. Again, we'll be happy to circulate details of Harry and anyone else involved in the meeting, and we'll kind of pick that up towards the end. But last by no means least is Dr. James Gavin. He is going to offer a very different insight, hopefully, in terms of an industry perspective. He'll tell us all about Evolution Therapeutics, which is a early stage biotechnology company that has spun out of Aston University, and hopefully maybe inspire some of the other researchers in the room to see what can be done with some of the EV work that's going on in the area. So, Jimmy, welcome to the network, and away you go.
Thanks a lot, Matt. Thanks for the intro. Just get this up. Can you all see that okay? Excellent. Right. So, yeah, thanks, Matt. Thanks everyone for sticking around and heard some really interesting talks. And as Matt said, hopefully it's a different perspective, this talk, where I can kind of tie the two sessions together. So it's quite good that I've come at the end. I've got coming from an academic background perspective and then actually forming a spin out and company. And really, I'm just going to give you an overview of my journey and how you could also do the same thing. So my name is James Gavin. I'm CEO of Evolution Therapeutics. It's a spin-out company from Aston University, which is trying to develop EV/nanoparticle-based therapeutics for the treatment of inflammatory conditions. So over the next 10 minutes, I'm going to talk about why you should also consider the commercial potential of your EV research. And if this is something that interests you, how can you take the first crucial steps? I'll touch on the reasons why commercializing EV research is not just an option, but actually a fantastic opportunity to create translatable real-world impact from your research. I'll also briefly speak about the how, what practical steps and support you can leverage to bring your research from the lab bench to the market. And to do this, I'm going to take you through my personal journey, from starting out as a PhD student in 2017 to leading a biotech spin-out in 2023, as someone with absolutely no background or experience in business. Really, this presentation is to emphasize that whether you're a PhD student, a postdoc, or a PI, if there's a commercial potential in your research, there are plenty of opportunities out there for you to do the same thing. So to rewind the clock a bit, In 2017, I started my PhD at Aston University, and even before COVID was a thing, the beginnings of my PhD were somewhat turbulent. But my saving grace at that time came in the form of a kind professor called Andrew Devitt, who adopted me into his group, and unfortunately for him, he's not been able to get rid of me ever since. So with Andrew's interest aligned to EVs and immunology, it steered me down the path of EV research. And during my PhD, I explored specific enzymes within EVs and their impact on the cellular microenvironment, specifically in a disease model of cystic fibrosis. These investigations yielded some intriguing insights into EV transport of active enzymes, and I took those skills that I learned during this time and transitioned into a postdoc position in 2020, where I shifted my focus to EVs from dying immune cells. And I must say that Dr. Ivana Milic, who's our keynote speaker after the break, had already done a lot of great work in this area. So I recommend staying around for her talk, and she'll delve into the science behind our technology. As such, I'm going to skip over the details, but just to say that similar to my PhD findings, EVs from dying immune cells also carried important molecules which could influence the cellular microenvironment, specifically in the control of inflammation. So with the support of the tech transfer team at Aston, and pushed by the fact that my funding was coming to an end and I would soon be out of a job, we asked the question, could this research be commercialized as a therapeutic treatment for inflammatory disease? What we envisaged was an idea to manufacture engineered EVs or lipid nanoparticles loaded with these immune modulating molecules that we had identified, and to use it as an advanced therapy for the treatment of diseases with chronic inflammation. But how would we go about translating our research into a therapy, and what would be the first steps? The answer for us came in the form of iCure, which stands for Innovation to Commercialisation of University Research, and in their words, the UK's leading early stage pre-accelerator programme, guiding researchers through the process of validating commercial potential. It's initially a 3-month programme funded by Innovate UK, but also in our case, BBSRC were involved, and it's designed to train and support teams led by early career researchers to determine whether there's a market for products or services that utilise their research. The team receives around £30,000 worth of funding to go out and get out of the lab and validate these commercially promising ideas in the marketplace via both digital testing and by speaking to 100+ experts from your respective field. Fortunately for me, my application to iCure was successful, and as soon as I had stepped onto the academic path, I was stepping off again, but this time to exotic places like Seattle, Barcelona and Telford. Okay, so not all were glamorous cities, but the funding gave me the opportunity to go to major events at these locations, which opened up the access to speaking to world-leading companies and having meaningful conversations with over 100 experts in the field. And from these conversations, I found out that we could manufacture and test our technology with the help of these companies. And it helped us to focus our attention on which clinical indication we should target first with our prospective therapy. As a point of note, the iCure program has expanded since the time I was involved, so they now have 4 different programs depending on your technology readiness level. So it really does open the door for anyone at any level to get involved. So what had I discovered during my 3 months of market discovery on the iCure? I found that non-healing wounds, for example, diabetic foot ulcers, have a high unmet medical need globally, and the therapeutic arsenal for wound healing is currently limited in scope. The burden to healthcare systems is immense. For example, the management of non-healing wounds in 2018 cost the NHS £5.6 billion, with 54 million community nurse visits, and patient quality of life remains extremely poor. And crucially, we could translate our research to manufacture these engineered EVs or lipid nanoparticles loaded with the immune modulating molecules we had identified to be used as an advanced therapy for the treatment of non-healing wounds. In time, we hope that this will prevent people with diabetic foot ulcers from undergoing limb amputation, free up more healthcare spaces, and give better overall quality of life to patients. So after 3 months of market discovery, you're asked to present your findings to a panel of experts from iCure at what they call an options roundabout. They then determine whether you need to return to the lab to do more research, license your technology, or actually form a spin-out company. So after the options roundabout, if you are recommended to form a spin-out company, which fortunately we were, you will receive a further 3 months of support to start setting up your business. For example, sorting cap tables and your IP and planning towards future investment. At the end of 3 months, you have the chance to apply for a grant, which is ring-fenced for the iCure applicants that were recommended to spin out. And this covers 100% of the costs for up to £300,000 to set up your company. So not exactly a bad idea. So after an intense period of grant writing, we submitted our iCure grant application, and we are fortunate enough to be awarded the funding from Innovate UK to set up our company. And thanks to the iCure programme, we'd already tested our idea in the marketplace, we had built the initial foundations of our business, and we now have the funding to make this all a reality. So one significant point of note is so far focused solely on iCure because that's the programme I took part in, but there are lots of great initiatives out there. For example, the West Midlands Combined Authority recently won a large sum of money to establish its own innovation accelerators. And you can find out more information if you're interested on the MediLink website stated. So that takes us up to the present, where we've now hit some key milestones, including submitting a patent for our technology, receiving further investment from Aston University to go alongside our Innovate grant to manufacture and test our lipid nanoparticle-based technology. And as a team with Professor Devitt and Dr. Milic, we've also secured a BBSRC grant to develop our technology in engineered EVs. In the future, we hope to advance our technology towards clinical trials from 2025 onwards, and hopefully translate our EV research into a real-world therapy which can change the course of patients' lives. I hope I've given you some food for thought and some inspiration to give it a go yourself. Of course, none of this would be possible without the team which has supported me throughout the entire process. So my gratitude to Andrew, Ivana, Matt and Luke. Thanks to all the funders and organisations that put these great programmes and initiatives together, and to the organisers today of the Midlands EV Network for putting on a terrific event. Finally, if you want to find out more about what we're doing at Evolution Therapeutics, the website address is there. You can also follow us on LinkedIn. And if you have any questions about iCure commercialisation or any complaints because you're from Telford, my email address is at the bottom and I'll be happy to answer them. Thank you all for listening.
It's brilliant. Thanks, Jimmy. Quite the ambassador for Telford as well, which is important. But yeah, I think it's a really inspiring to see you actually translate something out of the lab, and as with all of these presentations, you've made a lot of work look simple and smooth, and I know there's a lot more to it. So do contact Jimmy if you've got questions, particularly if you're PhD or early postdoc. There's a lot of good opportunities out there. And just as a further advert for that that program that Jimmy was talking about, there is a huge number of opportunities in and around the Midlands to support this type of activity. So again, any questions around that, use the network. That's what we're here for.